Good morning, and thank you for attending this meeting. So, as you know, there exists a large number of animal species living in groups or in societies in which individuals have the capacity to coordinate their actions and achieve a huge diversity of collective tasks. And this is commonly used to illustrate the concept of collective intelligence. It can be, for instance, the capacity of some species of ants to self-assemble and form chains that are used collectively to pull a large prey. And it can be the capacity of some termite species to build a huge and remarkably complex nest. It can also be the ability of many species of social insects to collectively choose the best food source or the optimal site to build a nest, or the ability of many species of birds or fish that lives in groups to coordinate their movements. And all this collective performance results from the interactions that take place between the individuals that make up this group. And during these interactions, the individual exchange some information, either directly or indirectly by means of traces that they leave in the environment. And this is these interactions that makes possible the coordination of individual activities. So when we try to understand the mechanisms that allow a group of organisms to collectively solve a problem, the first and essential step will be to decipher and characterize interactions that take place between individuals. And this is what has been done by an increasing number of research groups over the last 30 years. So today, we are in the beginning of having a fairly general idea of the types of interaction that allow groups of organisms to coordinate their actions. And we know in particular that these interactions are generally short range, which means that individuals only use local information about what happened in their environment. So the scale of this interaction is quite small in comparison to the scale of the structure built by the group or the structure that emerge at the group level. And also very often, individuals select the information to which they pay attention. So there is also some kind of information filtering. Individuals react to some information in their environment, for instance, some specific behavioral actions performed by the other group members, and they seem to completely ignore other kind of information. And we will see that information filtering not only allows individuals to efficiently coordinate their behaviors, but also each individual to minimize the cognitive load which is necessary to achieve the coordination. And this is what I would like to talk to you about today through the particular example of collective behavior of fish schools. So how can hundreds of fish move together in a coordinated way and behave as if they were the same single superorganism is one of the most captivating mysteries observable in nature. And as you can see, these groups of organisms can spontaneously adopt many different collective phases that can be rather disordered or much more organized. And for a long period of time, it was quite difficult to obtain accurate data on the collective movements of animal groups. So most of studies on these phenomena mainly relied on the analysis of mathematical models. In particular, in the 80s and 90s, many phenomenological models have been developed, and each of these models were based on specific individual interaction rules, and they were able to qualitatively reproduce the collective movements of schools of fish or flocks of birds that closely resemble those observed in nature. And for instance, if we look at some classical models of collective motion, such as the Aoki-Kusin model and also the Vicek model, they consider that within a group, each individual is influenced by all the neighbors that are located within a spatial neighborhood. And for instance, in the Vicek model, each individual aligns its direction of motion with the average direction of all individuals that are located in its neighborhood. However, the main problem remained the lack of experimental validation of the hypothesis upon which these models were based on, and particularly the hypothesis that concern the interaction rules at the individual scale, simply because very different interaction rules at individual scales are able to produce very similar collective dynamics. But now, for a few years, new methods that are based on machine learning algorithms have been developed to automatically track the trajectories and the behavior of animals moving in group. And these techniques have greatly improved the accuracy of the available data on social interaction, and they have also opened the way to build models of collective movement that are both quantitative and predictive. Now, the main difficulty in building such models lies in the close entanglement of the interactions between animals and their physical and social environments. And to solve this problem, some years ago, we have developed a method that consists in the first step to use experimental data to model the spontaneous movement of an isolated individual that doesn't encounter any physical obstacle on its way. And then in a second step, we use this model to measure the effect on the individual movements of its interaction with obstacle and with another individual which is present in its neighborhood. And then in a third step, we can validate the model of individual movement that include these interactions by comparing its prediction with the data collected from various experimental conditions, including experiments that were not used to build the model. And we can also try to understand how individuals combine information from multiple neighbors when swimming in a school. So we have successfully applied these methods to analyze and model the movement and the social interactions in several species of fish, and in particular in the ruminose tetra that you can see here. This is a small tropical freshwater fish whose average body length is about 30 millimeters, and that swims in groups in a highly synchronized and polarized manner. And like many other species of fish, these tetra perform a burst and coast type of swimming, which is characterized by sequences of sudden increase in speed, followed by mostly passive gliding periods. So if we look at the variation of the velocity with time, you can clearly see this succession of short acceleration phases followed by gliding phases during which the velocity decreases. And the short events during which a fish changes both its velocity and its direction of motion are called kicks. And this particular intermittent swimming mode makes it possible to analyze a fish trajectory as a series of discrete behavioral decisions in time and space. But the main interest of this swimming mode is that we can also use the spatial location where the kick have been performed by the fish to precisely identify the potential stimuli in the neighborhood that have elicited the behavioral response of the fish. And by accumulating several 10 hours of data on fish swimming alone or in pairs, we can identify the stimuli a fish reacts to. We can also measure the effect of these stimuli on the behavioral response of the fish. So let me just focus on the social interactions between two fish. What we found in this species is a combination of attraction and alignment, which are represented here respectively in red and blue, whose intensity depend basically on three parameters: the distance between the fish, the angular position of the fish, and although their relative orientation. So the strength of this attraction and alignment interaction depends on these three parameters. So for instance, if we look at the influence of the distance between two fish on the intensity of alignment and attraction, we can see that when the distance is small, let's say that 30 millimeters, which correspond to one body length, there is a short range repulsion. And then as the distance between the fish increases, we can see that the attraction, which is figured here in red, becomes more important and reaches a maximum value of around 200 millimeters, which corresponds to about 6 to 7 body lengths. And we can also see that the alignment in blue dominates the attraction up to 2.5 body lengths, while the attraction becomes dominant for larger distances. And as the distance between fish increases even more, we anticipate that the attraction will, of course, decrease. What we can also notice is a strong modulation of the behavioral response of a fish that depend on their relative position. And this is a direct consequence of their anisotropic perception of the environment. So this means that the interaction between two fish are asymmetric. In particular, the maximum amplitude of the alignment occurs when the neighboring fish is located on the front left or on the front right, and then the alignment vanishes as its position moves toward the back of the focal fish. And we can also see that the attraction is maximum when the other fish is located on the side of the focal fish, and it increases with its relative size, in particular when the neighbor is in front of the focal fish and with the same orientation of the focal fish, you can see that the attraction is minimum simply because its relative size from the point of view of the focal fish is also very small. Then it is quite easy to fit on these experimental data some mathematical functions that are represented here on this graph by the full lines to model the social interactions between fish. And the simulation of the model shows that these interactions can reproduce both qualitatively and quantitatively the movements of fish that we observe in the experiment. And in particular, you can see that the fish tend to stay close to the wall and also close to each other. But you can also see that the temporary leader, which is in the front, is much closer to the wall than the follower. And there is also a very good agreement between the model and the experiments if we consider the distance between fish and also the distribution of the orientation of the fish with regard to the wall. And then the big question is to understand how fish combine and integrate the interactions with many neighbors when they travel in group. And to answer these questions, we need to know for each fish and at each moment which neighbor influence its behavior. And more precisely, we need to know the number of neighbors fish pay attention to and also the strategy for selecting these neighbors. So to get answer to these questions, we perform a series of experiments in which groups of 5 fish we are moving in a circular tank, and we use the model that we developed for these species to investigate different hypotheses about the number of neighbors a fish pay attention to, and also the strategy to choose these neighbors. And then we have compared the simulation results for this experiment, for these different hypotheses, with what we observed in the experiments. So we have investigated 3 different strategies of interaction. The first strategy is based on the distance between individuals, so that fish interact with their k nearest neighbors, with k varying from 1 to 3. The second is a random strategy. The k neighbors are randomly sampled among the other fish, with k varying also from 1 to 3. And in the third strategy, the selected neighbors are those who have the largest influence on the instantaneous heading variation of the focal individual. And to measure the respective influence of each neighbor of a focal fish, we use the pairwise interaction function that we have implemented in the model with 2 fish. So that each time a fish performed a kick, we can precisely know which of its neighbor has the largest influence on its heading variation. So with all these interaction strategies, we can explore different ways for an individual to focus its attention on the most relevant stimuli in its neighborhood. I don't have time to describe in detail all the results that we got, but in summary, it clearly appears that the collective behavior that we observe in the experiments can only be reproduced by the model when each individual interacts with at least 2 of its neighbors, and no clear gain is obtained when individuals interact with a 3rd additional neighbor. And even more, when fish only interact with 1 neighbor, the most influential strategy leads to the best coordination, even when the group moves in an unbounded space. And this remains true for large groups. And here you can see on this simulation that groups of 10 and 20 individuals that only interact with their most influential neighbor remains cohesive. So now to summarize and conclude, Our result strongly suggests that each fish in a school must acquire only a minimal amount of information about the behavior of its neighbor to get a coordination of swimming at the group level. And this process may prevent information overload when the fish move in large groups. We know that in vertebrates, the midbrain and the forebrains are working in parallel to process the visual information and select the most salient stimuli that are the focus of attention. And in particular, the midbrain continuously monitors the environment for the relevant stimuli. So it is the primary site where the information about the neighbors is filtered. And then the forebrain selects those stimuli on which the fish focuses its attention. And our results suggest that fish do not have to pay attention to all their surrounding neighbors. They do not have to integrate all that information. They just have to react to the two most influential neighbors. And this is enough to ensure the coordination of swimming in a group. So finally, I would like to thank all these people that have been involved in this work. Liu Lei, Daniel Calovi, Valentin Lecheval, and Ramon Escobedo, who work in my group in Toulouse, and also Clément Cyr from the Laboratory of Theoretical Physics in Toulouse. Thank you very much for your attention.